All right, so we will get started. This is a Mushrooms 101 presentation by a fun guy. So I hope that everyone's ready for some corny puns. And as far as what to kind of expect tonight, we are going to kind of start off with what are fungi, why we should even care about them, and then we'll go into kind of basic mushroom anatomy and some of the broad categories of fungi as you kind of get out in the wild, start seeing some of these things, you can get very broad categories to them. This is a very, very complicated subject. So if you already have some familiarity with this, just know that this you can go pretty deep in with this. This is actually a presentation. This is a couple of presentations that I pieced together that is part of a much larger thing. So one, hopefully I keep to my time, but also two, we can go deep in each of these topics. So this is definitely just kind of a primer course, but at the very end, I will go through some resources that you can look into to learn more about this. Some of the people you can connect with to go on walks and guided hikes. And then we'll have plenty of time for questions as well. If you have questions throughout, try to throw it in the chat. I'll keep an eye on it, and then we'll have a nice question session at the end as well. But to start off, I do want to say, so is it fungi or is it fungi? I say fungi, but if you say fungi, that's correct. If you like the puns and you go with fungi, that's also correct. You can also say fungi. You can say fungi. All of these are correct. No specific one is more correct than the other. I've had people argue with me, especially those that want to be adamant that it's fungi or fungi, to which my response is, do you pronounce it fungus? Because it's usually not. So I have my personal preference. I think one is correct, but at the end of the day, all of these are correct. No specific one's better than the other. But now we'll actually get into what are fungi. And to start, we're going to need to go very, very broad. So this is a good tree of life example. And at each of these little intersections is representative of the last common ancestor before it split off, creating these different categories of bacteria, archaea, and then the eukaryotes over here. And we're going to go all the way to the right side of this representation of the tree of life. And in the green up here is kind of, we can think about plants. And then way down here, past amoebas, past some weird things here, are the two that I want you to focus on. These are the kingdoms, fungi and animalia, right next to each other. So fungi is actually pretty far removed from plants. This is something that was taught as plants up until like the 1980s. This was kind of pushed by an ecologist named Robert Whitaker, who really first proposed the five kingdoms that a lot people learned with Animalia, Plantae, Funga, Protista. So fungi are way more closely related to animals than they are to plants. And when we talk about fungi, for the purpose of this, we're going to really kind of only think about two main types. And this is how they are represented at a cellular level. So up first, we can think of the single-celled fungi, also known as yeasts. And this does include yeast that we use for fermentation. So that's a species called Saccharomyces cerevisiae. But it's also like 1,500 other species that exist as these single cellular organisms that reproduce partly by doing this weird budding thing that we see right here, where a little bud starts to form off of the main body. So that's the single-celled fungi. But what we generally see and think of as fungi is this one that it's multicellular. They're what we would say filamentous, or they make filaments that kind of weave together into a structure known as a mycelium. These individual filaments are cellular structures known as hyphae. So all these hyphae kind of weave together into a mycelium. And these mycelium can be pretty minute. And you've likely seen these, especially if you do a lot of gardening or you dig around or lift a log. Those white fibers, sometimes they're black, sometimes they've got some different colors to them. That's the mycelium of the fungus. That's the main body of this fungus. And it can be kind of tiny, might just be size of a quarter, or it can be massive. In fact, it's arguable that it is the largest organism in the world, the largest known organism in the world is a fungus in Oregon that's like 3.5 miles squared and possibly 35,000 tons and thousands of years old. It's just this massive mycelium that does put out roots of a type of fungus. It's a type of honey mushroom. Um, but for the most part, this is the true body. So when we think of mushrooms, the actual true fungus is this mycelium right here. The mushroom is the fruiting body. So we can kind of think of the mushroom as like a blueberry on a blueberry bush or an apple on an apple tree. So you can kind of think of it like the fruit. It's where part of the reproduction process will happen, and we'll get into that in a little bit. But I really want to hammer in that the fungus itself is these mycelial strands. And sometimes these are microscopic or we'll just never see them. They're so embedded in the substrate, whatever it's growing on, that we might not ever see it. Now, let's kind of really get into what is a fungus. Like what are the fun traits of fungi? And up first, they have cell walls, to which some might say, well, so do plants. In fact, so do some bacteria. But plants, their cell wall is primarily made of a structure known as cellulose, whereas fungi is made out of chitin. So it's a completely different structure. And so we should say that fungi have cell walls made of chitin. And chitin is a very interesting molecule. So it's a polysaccharide or a bunch of carbohydrates kind of chained together, linked together. And it's functionally similar to keratin, which is a fibrous protein that is in our nails and our hair. And chitin is also like, so it's structurally similar to cellulose and functionally similar to keratin. But chitin is found in most fungus, especially the ones that we'll be talking about. It's the main part of their cell wall, so the main thing that's given it some good structure. But chitin is also prevalent in the exoskeletons of insects and crustaceans. It's also found in the mouth parts of certain mollusks, like the radula or the teeth of snails or the beak of cephalopods. And we've even found it in vertebrates. So there is evidence of chitin in the epithelial layer of some fish scales. And even in larval salamanders, there seems to be some ability to make chitin in some of the appendages. So it is something that has persisted, this ability to synthesize chitin into the vertebrate lineage. They also, another interesting thing with fungi is they do absorptive nutrition through external digestion. And all this to kind of say that they put the digestive enzymes out into the world, let it dissolve whatever they're on, and soak up those nutrients. So we do absorptive nutrition through internal digestion. So that's kind of all happening in our stomach and throughout the rest of the intestines. But they do it by putting all those digestive enzymes out into the world. And we can kind of think of this in two main categories. You've got saprotrophs and biotrophs. Saprotrophs are going to be your decomposers. So these are the fungi that exist on dead material, primarily on wood. And this is a very important concept of the white rot and brown rot fungi, because fungi are so good. These saprotrophs, they're so good at decomposition. They are one of the primary reasons we can walk through the woods and not trip over a log every couple of feet, because they break down the wood. And when we think about wood, it's actually three compounds. There's cellulose, hemicellulose, and lignin, but we'll really just focus on the cellulose and the lignin. So lignin is the hard part of the wood. They're these big blocky compounds. They're so tightly compacted and linked so well that the carbon's trapped there. The primary way we can get the carbon out and to get the energy out of lignin is we have to light it on fire. So not much naturally breaks down lignin. And white rot does. So white rot fungi have compounds that can decompose lignin and cellulose. So in white rot embedded substrate, so this is like a branch that has some sort of white rot fungus in it, all you're left with are these little flaky linear strands of degraded cellulose. The lignin's been blasted away, releasing so much energy, so much carbon, and all that you got left is this flaky cellulose. But brown rot, less common, and brown rot really only degrades cellulose. And cellulose is what takes all those big blocky parts of lignin and kind of glues them together. So it's the glue that is holding the big blocky lignin together. And in brown rot, the fungus essentially just goes along these lines of cellulose, eating it away. Some of them will use hydrogen peroxide chemicals and just break this away, leaving the lignin behind. And that's when you end up with these logs that have just these big blocky parts. And that's just the lignin with all the cellulose taken away. So saprotrophs are one of the primary decomposers that you will encounter out in the wild versus fungi that are biotrophs. Biotrophs are fungi that get their digestion, get their nutrition from living material. And we can think of these as parasites. So there's plant pathogens. That honey mushroom that I mentioned, the massive 3.5 miles squared one, that is a parasite on living trees. Cordyceps is a genus of mushrooms that tend to parasitize insects and other invertebrates. So the fungus, it's actually like growing in this grasshopper until the fruiting bodies form out of the body. It's not going to end well for this insect. So these parasites usually lead to the death of whatever their substrate is. The other cool one that I think this is awesome. This is cedar apple rust. I had to throw this one in just because I think it's the craziest thing. So cedar apple rusts are actually a type of fungus that requires two different plants usually. In this case, this one needs an eastern red cedar and some sort of apple tree. So this could be actual apple tree or a crab apple tree, basically the genus Malus. And it doesn't necessarily need eastern red cedar, but something very close. And in eastern red cedar, when it's on the apple trees, which makes orange spots turn black, but then the little thing, when it gets to the cedar, it forms this brown gall that gets these little orange spots that start to grow out like Play-Doh. And these little gelatinous horns start forming. So rusts have crazy life cycles, but it is a parasite on these trees. And in fact, it can cause some serious crop failures for apple orchards. But biotrophs don't have to be parasites. We also have this idea of mutualism and mycorrhizal associations, which are symbiotic between the plants and the fungi. This is so prevalent that it is assumed to occur in roughly 90% of all land plants. 90% of all land plants form some sort of mycorrhizal association with fungi. In simple terms, the fungi is getting sugar from the plant, and the fungi helps kind of increase the uptake of water and mineral nutrients for the tree. So it's vastly increasing the surface area of the roots to really allow the tree to take in so much more. And there's probably a lot more going on with this as well. This is an interesting field of study, and fossil evidence of the arbuscular mycorrhizal association, which is a type of endo, and we'll talk about the endo and ecto in a second, but there's fossil evidence of this endomycorrhizal association dating back like 400 million years ago. But we do have two different types. So you have the endomycorrhizal association where these are the green is representing the fungus. This is actually going inside the root cells. We have a cross section of the root. The fungus grows in and then goes into the cell to exchange nutrients. And there's also ectomycorrhizal associations, not hard to say at all, that kind of weave their way in between the cells and exchange via the cell wall and cell membrane. So there are two main types. You can kind of break these apart into even further categories, though. So these two in particular, though, so the cell wall is made of chitin, and the fact that they do not photosynthesize, fungi do not photosynthesize, they have to digest and obtain their nutrition from another organism, really helps tie in the fact that fungi and animals are closely related because chitin is prevalent in the animal kingdom, especially in insects and other arthropods. And the fact that the digestion has to occur. There is no photosynthesis like in the plant kingdom. To continue on, though, another trait that helps define fungi is they do reproduce via spores. Now, the next slide, again, we can go as complicated as we want to with any of these topics. This is going to be a broad overview of the reproductive cycle. And we start out, we got these mycelium. Conditions are just right. They decide to make a fruiting body. So this fruiting body starts up, got this little tiny structure, goes in, eventually unfurls into what we would think of as a mushroom, and we have gills on the undersurface here. So these gills, that's where the spores are going to be produced. Don't worry about the terminology that'll show up on this slide. These gills have a bunch of these microscopic structures. Each of those little circles is a spore. So these structures are producing the spores. They're getting ready, and eventually we generally have 4 spores per structure, and those spores get released. In this instance, it's going to get released and it's going to get blown by the wind. So this is a wind dispersal mushroom, and those spores go out, and sometimes, I mean, it really is as far as the wind will take it. But some studies have shown that these can travel thousands of kilometers, if, especially in the right condition. Some studies were done with wildfire smoke and finding fungal spores within the wildfire smoke kilometers away from where the fire started. And eventually they will land on the ground. So we have 2 spores that are now going to germinate and start creating those hyphae. So the hyphae, the filamentous multicellular main part of the fungus that makes the mycelium, starts to form. So we had a spore here, and then let's say this spore was hundreds of feet over or something like that. They have to eventually find each other. The 2 mycelium have to join to make this next stage, this plasmogamy stage, which we won't go into much detail, but essentially at this point they have 2 nuclei in them. And that is kind of where the sexual reproduction can start. Many fungi can do asexual reproduction, but I just want to put this out here that this is incredibly complicated. There is a lot of different like compatibility. These mycelia have to be compatible, and it's like plus minus, but also there's a lot of different things that make them have to be compatible to finally make these mushrooms. And that's what's insane, that all this is just happening right now. The fungi also have very unique cellular features, and especially in some of the what we would call higher orders. These are structures like clamp connections, which is prevalent in something we'll talk about called basidiomycetes, which is where we get the mushrooms that we think about. But these clamp connections are, it's unique to them. I mean, they get these to allow this stage where you have 2 nuclei from 2 different cells existing in the same cell. This is all kind of part of the reproductive cycle. But fungi do have traits that make them unique, as well as some that overlap, like reproduction via spores is prevalent in other things, including plants. But all these together kind of define fungi. So now that we have hopefully a base level of what fungi are, I'd say the big key takeaways from this is the fact that they do not photosynthesize. They have to get their nutrients from something else, and that's either decomposition or a living organism. And they have chitin, and that chitin is also prevalent in insects. So now some might ask, why should we care? Why are fungi important? Well, one, they drastically speed up the carbon cycle. Well, again, without fungi, we wouldn't be able to walk in the woods. That white rot is just so extremely effective at decomposition, blasting away the lignin, exposing the cellulose to be quickly converted into glucose that's used for food. Other things can also break down cellulose a lot easier. So it's such a crucial part of the carbon cycle. There is also medical significance to fungi. About 30 species can cause serious illness in humans, like aspergillus. There's also some good medicine. In fact, penicillin, one of the first antibiotics we ever had, came from fungus. And this is a field that is constantly being researched. There are a lot of um, folk remedies that involved fungi that are turning out to be very viable. So we are finding, for instance, a mushroom that we'll see later in the slide here, turkey tails. It can get prescribed by veterinarians to reduce the size of tumors, just certain types of tumors in dogs. And it had been used as a tincture for a long time, as like a probiotic. But now we're starting to see that some of these compounds do have serious medical impacts. There are also significant plant and wildlife pathogens within the fungi kingdom. Wheat stem rust on the left here saps nutrients from wheat, cause the stems to fall over, shrivels the grain, overall desiccation of the plant. So it is a serious plant pathogen for the grain industry. Over on the right, some of you may already be familiar with white nose syndrome. This is a fun species to pronounce. It's Pseudogymnascus destructans. But this is a fungus that was first found in the US, at least in 2007, and has since spread to 40 of the states. It's infected several species of bats, resulting in some die off events of 90%. 90% of, I think, the northern long-eared bat, little brown bat, these populations have been decimated by this fungal pathogen. Chestnut trees, the chestnut blight was a fungus. The American elm, Dutch elm disease. These are very important plant pathogens that have impact our ecosystem. So understanding fungi, what their role is ecologically, is very important whenever we have these new novel pathogens show up. Fungi are also a crucial food source for us, and I am not talking about these button mushrooms that you can get from the grocery store, although that is an important part of diet to many people, and going out and foraging mushrooms is also a very important pastime for many people, both culturally and just personally. But what I'm talking about are these guys. These guys, this is yeast, specifically Saccharomyces cerevisiae. This is the yeast that we have domesticated for thousands of years, giving us bread, cheese, beer, wine, soy sauce, tofu. All these fermented foods came from yeast, which is a fungus. So that's one reason why we should care about it. But really, there's only one thing that should be that important for everybody. And that is, why do I care about fungi? And this might be why you end up caring about fungi too. The most important thing to me is I just think they're neat. I mean, they're just so great. Look at these things. This is insane. This is just something. Each one of these I just randomly happened upon on an average walk. Birds are great. Many of you know me. I'm a big birder. I love plants too. I love taking pictures of flowers. I love seeing all the trees in the wintertime. But fungi. This is where I really get passionate. I mean, where else are you going to see these colors and these growth forms outside of a sci-fi novel? I absolutely adore fungi. And with that, I will pause for questions here. I don't know if anyone has any. I'll take a quick little break. I think you guys. Oh, you can't unmute. You can unmute yourself now if you needed to. But any questions before we move on? Well, we will continue on the way. Oop, I think I saw a chat. Yeah, sorry, there is a lot of material. So I will send the recording and I'll give you a lot of good resources to look in. When people say you can pick a mushroom and won't, oh, this is a good question, Heidi. All right, so when people say you can pick a mushroom and it won't hurt the population, how can that be when spores are prevented? That is a good question. So the idea that you can pick a mushroom, so there's a lot of ideas where you go out and you pluck a mushroom off the ground. Let's put this guy's back on here. So let's say we go out and we see this cool mushroom called the old man of the woods. You want to collect it for your own collection to identify it. You pick it off. There's argument to be made that you essentially have just plucked a blueberry. So if you pluck a blueberry from the tree or from the shrub, it really isn't going to impact that blueberry's success overall. However, did you just take all of its spore potential? Probably. I mean, there's a good significance of those spores that could have been produced, but a vast majority of those spores have already released. The moment that mushroom has come out, it was under the right conditions. As soon as that thing kind of unfurled, you've got these spores out. There will be more spores, but the vast majority of them have kind of already gotten released. And the other part of this is if I come across one of these, most of the time I'm find three, four, five more nearby. And I'm not going to collect, even if it's a foraging thing. So I'm out just trying to collect food for the table, or I'm just out collecting to identify. There is the ethics of not taking more than you need. Make sure to leave some in the wild. You always want to just keep some there. So there is this idea that you could be taking them. But when we get to the part about what to bring when you go mushrooming, there's a few tools that you really have to bring with you. And one of them is an adorable wicker basket. And the reason why is it kind of helps air it out. But now you have this idea that we're helping the mushroom. So let's say we put in a bunch of boletes that we're going to eat for later into this nice wicker basket. It's keeping them from getting all gross and wet. But also, those wicker baskets have nice holes in them for the spores to go through. So now I'm tracking these spores all through the forest, and I'm doing a better job of spreading these spores than the mushroom could have ever hoped for. So there is a question for it, and I don't necessarily have the right answer. Um, so there's a roundabout way of saying it's kind of a murky situation anytime you talk about ethics. And then we got the picture reminds me of the seafloor. Oh, well, that's a good thing to hold on to there. So everyone think about what the orange one reminds you of. And then in the world of color, there's the lesson that should we stay away from the bright reds, yellow, true or false, or is it fungi specific? So there are no general rules that are safe when it comes to mushrooms. Like if you're eating them, you can touch them, you can look at them, you can even smell them. That is fine. Do not eat mushrooms, especially unless you yourself are confident on its ID. This is something that I stress on all my walks. Even if I tell you, oh yeah, that is chicken of the woods, that's the best, that's like my favorite mushroom to eat. It's really hard to confuse it with anything. Don't take my word for it. You yourself have to be sure. You have to work through the keys. You have to make sure before you eat any mushroom. There are no good general rules for like if it's brightly orange, it's toxic. If it's dull brown, it's not. In fact, one of the most deadliest toxic ones is just white. And you can safely touch even that one that would kill you outright. You can touch it. You can pick it up. It's you have to ingest it for these toxins to be metabolized. So do always kind of go with that little bit of caution when handling these. Just don't eat them. But the colors themselves don't necessarily signify the ones that are toxic. In fact, um, the bright orange chicken of the woods is a very, very delicious edible one. All right. Any other questions? Did I get them all? All right. Well, we will continue on. Now we'll talk about mushrooms in particular, and then I'm gonna take a hard right and show you that not all the well, I mean, we already spoiler alert, not all fungi will look like mushrooms. But we will talk about the mushroom. So basic mushroom anatomy. Whenever you flip open a field guide, you'll come across these terms, and it's good to go ahead and get them in your head. Mycologists, I always joke, wanted to be different than the botanists. So everything that could have been called the same thing in plants, they had to come up with a different name. But we do have at the top here the pileus or playus. This is the cap. So the cap of the mushroom, the top of that mushroom. Oop, gotta check. Uh, um, I will answer that one in a second, Ben. We'll get to that one in when we get to the birds because I've got a good spot to talk about that one. But the cap itself is up at the top. Occasionally, mushrooms will also have a uh, ring or an annulus. This is actually as the mushroom comes up, that annulus is attached to the underside of the cap. And as the cap kind of unfurls, that ring breaks off and is stuck still on stalk, also known as the stipe. So the ring is not always prevalent or present, but if it is present, that is a good field mark. So always make note of if there is a ring and about how big or wide it is. But these are the three main parts of the mushroom. We have the cap up top, the ring, and the stalk. Then we occasionally have different components on top of that cap. Does it have scales? Does it have striping? The scales are also kind of a remnant, oftentimes, of as it comes out, there's an additional little structure that makes it look like an egg and it rips away, and some of that veil that was left stays on the top. So any of these features, you're going to start to find that identifying mushrooms comes to so many little minute details, so many nuances on different things that you have to observe everything. If it doesn't matter how insignificant it seems to be, that could be the difference between identifying it or not. But then on the underside, we will think about the hymenophore or the spore-bearing tissue layer. So this is where the hymenium or the spore-bearing tissue layer occurs. And this is a complicated word, but this is just what does it look like on the underside of the cap? Okay. So the hymenophore, what does it look like under the cap? We have four main categories that we'll think about when it comes to the mushroom hymenophore. We have gilled mushrooms. So think about the ones that you can buy at the store, the little button mushrooms. They have those little gills on the underside. They could be toothed, so they look like little stalactites hanging down. They could be pored, so they're little holes on the underside. Or in some cases, they're just smooth. But in all of these cases, if we got a microscope out, took a look, we would see the spore-bearing structures deep within, embedded in the hymenium or the tissue, like the spore-bearing tissue layer. And this whole structure where that hymenium is occurring is known as the hymenophore. But you can just think about it as the underside of the cap. So just to refresh real quick, we got cap, we got stipe, and occasionally we'll have other structures like a ring or scales, but big thing, cap, stipe, and the hymenophore, which can be gilled, toothed, pored, or smooth. Whenever you get into I'll have a few field guides to recommend and some resources and whatnot, but you're going to start to see that mycologists also love adjectives. They love them. We have a lot of words for a lot of different things, especially just the cap shape or the way the gills form. So the way the gills attach to the stipe can also be a very significant thing. So you'll see a lot of times, don't write this down, they'll be in any good field guide. The gill attachment or the morphology of the cap can be very important. So are they decurrent, where your gill, so the blue parts of the gill, this is a cross section of a mushroom, but the gill runs down the stipe to some degree? Or are they completely free? So your gill goes all the way back up to the cap and never touches the stipe? Or is it somewhere in between? It's subdecurrent, or do they kind of pinch upwards right before the stipe, and they do connect to the stipe, but they're a little bit more pinched? And then the shape of the mushroom, the cap, can be also important. Is it trumpet shaped? Is it flat, depressed, conical, convex? Is it umbonate, where you got this little snub at the top, this little knob at the top? It's all very important. And when we think about these hymenophores, I mentioned the spores. So why care about these spores if they're microscopic? Well, you can also obtain what's called a spore print from these. So whenever we cut, let's say that old man of the woods that we talked about, or these little red ones, this is actually this here. If you go out looking for mushrooms now, this is a good time to see these. This has probably my favorite common name ever. This is the bleeding fairy helmet. It's a type of mycena mushroom, but again, bleeding fairy helmet. It actually will secrete out this little red wine, dried blood colored red. That'll stain your fingers for a little bit. But let's say we grabbed that. We took this cap. We could put it on a piece of paper and leave it overnight, and it will drop spores throughout the night. Especially we put like a bowl over the top, maybe even just a tiny drop of water on the top of the cap to simulate a little recent rainfall, increase the humidity under there. And the whole time it's going to be dropping down those microscopic spores. And as time goes by, those spores are going to pile up on top of each other. And before you know it, we will be able to see those spores all collectively together. And the color of the spore print can be very important for ID. Just knowing, is it white or brown, can separate two very similar looking mushrooms. So a lot of times some people will just collect mushrooms to make artwork. This is the thing when it comes to fungi, you need to find out what you want out of this. Is it to forage? Is it to eat mushrooms? Is it, personally, I just love to go out, photograph them, and try to identify them. I find it very challenging and rewarding to identify them. Um, Frustrating as well, a lot of dead ends. But some people just collect these spore prints and make art from them. They're very unique colors. You can preserve them afterwards with single-sided tape, or I've heard hairspray also really works. And for collection wise, you can even find these cool little things online. So this is out of the Mycological Society out of Toronto, actually just has these on their website for you to print. But you have half black, half white. They've got a little measuring in here. So you put the cap right here, and this way, if it's white spores, or mycological pink, which is like real pale pink, um, it'll show up really nice on the black, versus all the other colors will show up really nice on the white. You got some information on where you collected it. So aluminum foil, glass slides also works, but spores can be a very important component whenever identifying a mushroom, but they're all coming from the hymenophore. So you kind of have to make sure that that part's the part that's hanging down. And I want to remind you all that the primary fungi is, again, this mycelium. This mushroom right here, this is the fruiting body. But that little spot, that's where all the spores are going to be formed, on these gills. The main organism just in this substrate, living its life, going all over. All right. Now we'll get into the basic types of fungi. This is a huge kingdom. We have like, oh geez, I can't remember. I think 150,000 described species, and probably 1,200 are described every year. 12,000 new species every year. The estimate for like how many species of fungi are probably in the world are, you got like 1.5 million over on this side of the spectrum, to an like 10 million over on this side. I think the average is somewhere around like 3.5 million species is what we expect. So it's a big category. I will start this off with a fun comic that was shared with me. So if you haven't checked out, Rosemary Moscow does some awesome nature writing and comics. and this is her definition of types of mushrooms. So we will actually be going through all these. We got the morel, that's creepy but tasty. You got the little brown mushroom, boring but deadly. This is actually deadly Galerina. We got the jiggles if you poke it, jelly fungi, just eggs. These, I actually have a photo of one of these in the slides. They are deeply disconcerting. It looks like they're just weeping blood. And tree nose, coral fungus. When we get to the one that's censored, you'll actually probably see why it was censored in this photo. We'll talk about that one in some more detail. So this is kind of what we're about to go through, though. Come up with your own fun names for them. It's great. This is the phylogeny. So this is the tree for fungi. We have a lot of different large categories, including like Chytridiomycete. Chytridiomycetes are aquatic. This is actually, if you remember the big frog fungal disease, BD, and then there's one in Europe called B. sal. This is the Chytridiids. But what we're going to focus on is down here, Dicaria. So that's actually the species that make that two nuclei cycle at some point. And we're really only focusing on these two. So Basidiomycota and Ascomycota. So Basidia and Asco. We're going to start with Ascomycetes. And that means Asco fungi or sac fungi. And both of these, so the ascomycetes and the psidiomycetes, they're named for this microscopic structure that bears the spores. So where the spores are formed in ascomycetes looks like a sac. So this is just a big old sac with one opening over here, and these black dots are the spores. So in sac fungi, the spores are in a sac. And this is a weird group. Little There's about 64,000 species. And that includes morels. So this is where a lot of people get excited about this group. This morel, fun story, was in Alaska. This thing was huge. I was visiting a family member and we were about to head out to the airport. Like I was leaving in 10 minutes and she was like, "Hey, Nick, I hate to do this, but do you mind going in the backyard and picking up all the dog toys?" She really couldn't get on the hillside. So I go out there and start picking one up. And this morel is like this big. And I'm sitting there staring at it, having to go to the airport in like 5 minutes, get in the car and leave. I'm like, what am I supposed to do with this? And I tell her about it. She's like, "Oh yeah, those pop up in the backyard all the time." I'm like, "Why? Why would you not tell me about?" Anyway, this is a delicious mushroom. I ended up leaving that one. I didn't want to risk bringing it to TSA. I don't know what was going to happen. But you also get some cool little cup fungi. So a lot of ascomycetes have cup-like formations. These are red rubber cups is a common name for them. Lichen, which we'll talk about towards the end here, about 98% of lichens have an ascomycete as the fungal component. Shadowing for learning when we get to lichen, but it's not just fungi in there. But of the fungi components, about 98% of lichen are ascomycetes. And the Saccharomyces that we talked about that we use for primary fermentation agent, like baker's yeast, is an ascomycete. There are basidiomycete yeasts, but the yeast that we care about is an ascomycete. One of the fun ones that I found fairly recently is this guy. So I'm hiking through the woods, and I was actually like out trying to look for mushrooms just to make a bunch of iNaturalist observations, and I see this orange off in the distance, like, "Oh, cool, what's this?" And I get closer, and I was like, "Ah, dang it!" And I thought it was just discarded orange peel, because you get that on the trails all the time. You'll just see like someone takes one of those small oranges, throws the orange peel off to the side, and I was like, "Ah, that's all this is." And then I like look closer. Wait, this is a mushroom. So I take some photos, and I get to my field guide, and I figure out what it is. And the common name for this is orange peel fungus. And I was like, well, that's appropriate. But yeah, you might see this one around. I saw it growing on dirt, but it can also kind of grow on some shallow leaf litter. But it looks just like an orange peel. So these are the ascomycetes. These are kind of the weird ones. Tend to be cup shaped. You do have some unique looking things in there, but for the most part, they're rubber cups and morels. And then all the lichen. Now we're going to get into the big group, the diverse group that we see the most of. and that is the basidiomycetes. These are the club fungi. So the basidium, or plural basidia, is this structure right here. This is supposed to be club shaped, and then usually four little points up at the top will have one spore each. So this microscopic feature kind of defines our group here, basidiomycetes, or the club fungi. And now we're going to go into the broad categories on how you can kind of think about these. The thing is, genetically, a lot of these categories don't hold up. So just because we're going to talk about polypores and just because we're going to talk about jelly fungi does not mean that all jelly fungi or all polypores shared a common ancestor. In fact, that's very rarely the case in these broad categories. This can get very complex very quick, but we're just going to talk about what they look like, okay? It's a fascinating field. It's constantly evolving. It can be a little discouraging, but don't let it be. Whenever you find out that the way we thought about things is changing, it's just fun. We're just learning more about these. But what we'll look at is just morphologically, like what do these things look like? Up first, we have the bracket or the shelf fungi. These tend to form on trunks or branches. They're mostly saprophytes, so they're feeding on dead wood. They're one of the most prevalent wood rot fungi. So the growth form, again, it developed multiple times across multiple families and orders. In fact, like some polypores, which have the little holes, the pored surfaces are actually really closely related to some guild mushrooms. Again, gets complicated, but this is kind of where we talk about bracket fungi. So we've got these nice shelves or brackets hanging off of, on this case, this is a black locust tree, and this mushroom really only occurs on black locust trees. And it's the cracked cap polypore, or the mossy cracked cap polypore, only really comes up on this tree. Or we have Ganoderma suga. Don't worry too much about these names. The hemlock varnish cap looks like someone kind of varnished the top here. But this also includes turkey tails. So here's a little showcase of one of the polypores. The fairly common one. The problem is there's a lot of lookalikes. So you do want to learn to ID turkey tails versus some of the other ones like the false turkey tail, which is a type of crust fungus, or Trichaptum biformes, the violet toothed polypore, which has looks like little teeth instead, and the margin is purple. But in turkey tails, the way you can start looking for this is go on a good mushroom walk with anyone, and this is usually one of the first ones you'll get to learn. But there are fine velvety hairs on the upper surface. So this is the upper surface. The colored surface is what will be showing up to you on a log. And it's striated, so hence the name turkey tails. Kind of looks like a fanned out turkey tail. But you have all these different striations of varying colors or shades of brown, and then a margin of white. And if we took one of these off and looked at the underside, or flipped the log to look at the underside, it will be bright white. So there will be no striations. And then this photo is actually zoomed in quite a bit. And there are microscopic pores, well, not microscopic, near microscopic pores, tiny, tiny holes. You'll need a hand lens to see these. And essentially, the way you can tell this from other closely related species, if you got to the point that it's got the velvety hairs and you flipped over, it's white, there's about like 4 to 8 holes per millimeter versus like 1 to 3. So 1 to 3, the holes are going to be pretty easy to see even with just your eyes. But these, you essentially need a hand lens or zoom in on a photo to see them. So turkey tail is a type of polypore. Polypore is another name for shelf fungus or bracket fungi that have these pores. So the polypore, you've got a lot of different pores. Another large group that's kind of weird, the jelly fungi. Jelly fungi include witch's butter, It's a fun common name for this one. It also includes some ear fungi. Some of you might have seen this, tend to see this after a rainstorm with heavy winds. You'll go out and there'll be a lot of branches falling on the ground, and some of them are covered in this stuff. And you like pick it up, and they're like soggy. They're flapping over the side. These actually will dry up and turn into little tiny black spots when it's drought season, and then they swell up with water after a rain event. But these are known as amber jelly rolls. So the jelly fungi, just weird. These are the ones that you poke it, they jiggle. The interesting thing on these is you have to really look at the basidium to identify them. So sometimes when you're identifying mushrooms, you have to look at microscopic characteristics. And the jelly fungi have weird looking basidia. So instead of being that club shape, they'll sometimes be like tuning forked, or instead of branching all at the top, they branch off at the side. Jelly fungi can get kind of weird. Coral fungi. So someone had mentioned earlier that the orange one looked like coral when you're scuba diving. There's a whole like bunch of different coral fungi out there coming in all sorts of amazing colors. So the purple ones I've seen in the southern Appalachias. You can see these growing around this time of year. These white ones you'll see sometimes on logs. They're just a cool group. Pretty hard to ID though. So the spores are on the side here. So you'd actually just kind of lay this down and it would drop spores. Tend to need to look at the spores under a microscope to help identify this to species for some of these. We also have toothed fungi. So this includes the toothed mushrooms, but also things like lion's mane right here, or the mealy tooth, which has the crazy, like just leaking out red water. So there's a chemical in here that'll kind of stain the water red. Very, very odd to see in the field. So tooth fungi, lion's mane is one that a lot of people seek for foraging. It's actually probably a complex of a lot of different species. These tend to be found on living oak and beech trees, and they're readily identified because they form like a single clump of dangling spines that often are like over a centimeter in length, each of those spines, but they just look like frozen waterfalls. They're so beautiful. Another weird group is the gastromycetes. So we're still in basidiomycetes. Gastromycetes is actually kind of this obsolete classification, but one of the books I'll share really leaned into this group, and I think it's valid. In fact, when I took my mycology course, I was still taught gastromycetes just because it makes sense. So gastromycetes are the ones that their spores are produced internally. Whereas on the guild mushrooms and all the mushrooms we've talked about so far, they're external structures. Those basidia are on the outside, and they release the spores like ballistically. They go out and pop off and go into the air. The gastromycetes, they form them inside in some way. And we're going to see some of those in a second. And then they're passively released. Usually they require some other organism to help them out. So gastromycete isn't a thing that is evolutionarily appropriate because all of these kind of evolved multiple times across the same across multiple different families. But it's a good way to think about it broadly. The one of the gastromycetes are the like earth balls or earth stars or the puff balls. These are the ones that kind of on the inside. So we'll take a look at this earth ball here. We can think about this for the puff ball too. So the puff balls, when this was fresh, if we cut on the inside, it'll be completely white on the inside. And this structure in the middle is essentially normal, like structural tissue. So it's just the chitin until some certain trigger happens and it starts self-destructing all of this tissue in the middle called a gleba, Gleba. So the gleba is at the center here and it starts self-destructing into spore tissue. And now this whole thing is full of spore tissue that degrades down. So even in the puffball that was white on the center, now it's kind of turned brown until at some point a little hole opens. And if you haven't seen these, I encourage you to start looking around for them. They're real fun. You go up, you tap this, and a puff of spores comes out. And this is how they spread. So as a deer walks through and tromps on this, puff. As a chipmunk comes and investigates on it, takes a bite, puff. And then the chipmunk's also covered in some spores that'll hope it'll probably end up wiping on some other things. Earth stars will do the same thing, but you have this additional structure on the outside that gives it the name Earth star. Earth balls are very, very firm. They do eventually degrade. Puffballs generally are looked for foraging because they can be pretty safe. They're just you cut them, they're white on the inside, you're good to go. The main one that people get poisoned by is one of the earth balls, which is confusing when you hear the common name. I know, I'm sorry. But the pig skin poison puffball. So this is actually gets confused as a puffball. If people don't cut it open and see that it's black on the inside, it can poison them. But even just like a puffball, this eventually degrades. And so you have a bunch of spore material on the inside here. Then we have one of my favorite ones. So these are birds' nest fungi. These are unique. I mean, this is crazy. I encourage you check out in your mulch bed. You'll probably have some of these just growing right on the mulch chips. But these little structures here, this is a fungus. This is a mushroom. So you've got this cup. When it first forms, it'll be covered in a drum skin that peels away, revealing these little eggs on the inside. So these eggs are called peridioles. So peridial, and they're attached by a tiny little cord. And this is where it gets crazy. So this peridial is where all the spores are. So they're packed in there, this covered in this tough outer shell. And in a rainstorm, a rain droplet is going to fall into this funnel and hit with enough force that kind of causes these to shoot out. But as they shoot out, this little structure unfurls and you have a cord called a funicular cord. So this cord unfurls from the bottom of the spore packet and it's sticky. So this shoots out and ends up kind of wrapping around vegetation. Now, why? Well, this spore packet needs some like scarification, like a seed, to germinate, to release those spores. So what happens is this is now at the top of some sort of herbaceous plant, and a deer is going to come by or a rabbit is going to come by and munch on this, digest that little packet, and now those spores come out the other end with one, probably a good substrate with the deer rabbit feces, but also the scarification has broken apart that peridial outer shell, releasing the spores on the inside. It's nuts. Anyway, birds, nest fungi, check them out in your local mulch. I highly encourage you to look at them. They're not hurting anything. Then we got weird ones. Um, this is the one where I probably could have censored some of these. The stinkhorns. So we're still with the gastromycetes. They're still making spores on the inside, but then they get weird with theirs. They smell. So this mess on the outside is kind of sticky, slimy, and stinks. A lot of time it smells like rotten meat, and that brings in flies. So flies will come in attracted to the slime smell, and they start lapping it all up, getting the spore juice all over them, fly off, spreading the spores that are still all over their body hairs. So they need insects to disperse their spores most of the time. We have some really weird ones. The one of the genuses is just phallus. So there's some odd shapes, very disconcerting shapes. If you ever see any of these, I think they're pretty insane looking. They look like little alien eggs. But like this one, those are flies on the inside. So all that gleba, so the tissue that's self-destructed into spore tissue, is all on the inside covered with stinkhorns in a forest near you. Now, again, still in the basidiomycetes, but we are into the mushrooms. These are the ones that you generally think of when you think of mushrooms. So the gilled ones, you got your chanterelles, you got your boletes. Boletes also could be think you could think of these as like stalked polypores. So boletes are the ones that have the little holes on the underside. So that is where your spore tissue is being made. A lot of boletes also have some cool oxidation events. So this is a bolete. So this is the underside, that hymenophore. And what has happened is one, someone stepped on it right here, and then realizing, oh, look, it's turning blue. So it was naturally yellow, but as pressure was applied, it turned blue. They carved a message to their mother. So these will tend to oxidize if they're exposed to air or damage. So you can press on them, they'll bruise, slice into them, and they might instantly turn blue, or they might take a little bit of time and then turn blue. So boletes, you got the pored holes. Sometimes they might oxidize, sometimes they might not. Um, they tend to occur in like scattered amounts. So you usually have more than one. And then you also have chanterelles. These This is where you can kind of, this is more smooth or ridged than gilled. This is where you kind of get into a little bit of confusing territory. So chanterelles don't have true gills. Instead, it's like the stipe and the cap are just ridged where they meet. And these also tend to have very pleasant odors. Will smell like apricots. They, again, usually occur very gregariously. So you have a bunch of them scattered all over one hillside. And then another group here is the agarics or guild mushrooms. And this, again, we're in mushroom 101. So I just want to go over the broad groups, but this is where things can get complicated. This is where you really need to do spore prints. You need to take note of where it's growing. We'll talk about that in the resource section. So was it growing on wood? Was it growing near a oak tree or under a pine tree? Did you dig it up? We'll talk about why that's important. But gilled mushrooms, and we have those gills. Some of them actually secrete a little like milky latex when you cut them. Sometimes they'll have the caps on it or the scales on top. So those are our fungi broad categories. Now we are going into the not quite fungi. Just two more things here, just to make sure that you know they're separate. First up, slime molds. These are not fungi. This is actually an informal name. They are not molds. They are unrelated organisms. The slime molds that we tend to see are these, what we call plasmodial slime molds. And it's basically like one large cell with thousands of nuclei and no walls, just kind of roaming around until they need to create spores. And that's when you get these weird structures. So this one is the chocolate slime mold. So it's got these little stalks that come down, and this is some spore tissue. Some of you might have seen this one. It gets called scrambled egg slime mold or dog vomit slime mold. They'll show up on mulch. So this is actually moving. So this whole creature is just kind of rolling over itself along branches and leaves looking for microorganisms. So it's not hurting anything. It's just lapping up a bunch of tiny little microorganisms. And then when it's ready to sporify, it kind of condenses into one structure, turns brown and brittle and then dusty. And cotton candy slime mold. So slime molds are not fungi. They warrant their own program all on their own. And then lichen. Lichen are not fungi. They're actually not a single organism. So this is a symbiotic relationship. You have a fungus and a photobiont. So something that can photosynthesize. So the fungus is multicellular, creating most of the structure. And then the photobiont, which is usually an algae or a cyanobacteria, are the single cell organisms, and there's thousands of them all inside here. And for a while, like when I was taught lichen in school, that I was taught that it was mutualistic. So the photobiont produces sugar, the fungus gets the sugar, and in return, it's creating a nice home and protecting the algae from the outside. Like the ones that occur on rock faces usually get exposed to a lot of UV radiation, and the fungal pigments help protect the algae from like oversaturating in the UV radiation. I wouldn't say it's mutualistic because the only way the fungus can actually obtain the sugar is by penetrating the cell of the single cell organism and sucking it out. So the only reason this works is because the algae or the cyanobacteria reproduce so rapidly that they're kind of constantly, there's no net loss of photobiont. And that just doesn't sound very mutualistic to me. But anyway, it is symbiotic. So you have a fungal component, and 98% of the time it is an ascomycete fungal component and then some sort of photobiont. And what's interesting is the photobiont can be found wild out in nature. Just in streams, we can find the same algae that's in this middle lichen. But majority of the fungal components do not exist on their own. They do not naturally occur in the wild. In lab settings, we've isolated them, but that's it. So I think that's kind of interesting. The fungus really needs the photobiont, but the photobiont doesn't necessarily need the fungus. And when it comes to lichen, you can kind of categorize them in three different components. You have crustose, which are just the crusty ones. They're almost two-dimensional. They are just a crust on rock or bark. Foliose, over here on the left, you get a little bit more three-dimensional. It looks like foliage, so it looks like leaves coming off. And then things get erratic with the fruticose. These are the ones like bushy beard lichen, which just looks like a bushy beard, but green. All right. And then we just got a little bit of resources that I wanted to go through, and then time for more questions. So the question might be, well, you know what? I want to go look. Well, what should I bring? First off, you definitely want a notepad and a camera. You want to take notes. You want to constantly think like, okay, I found this mushroom. Where did I find it? Was it by this tree? What's the weather like? Has it rained recently? Anything that you could possibly think of, make a note of this. You also want to bring aluminum foil or paper bags, especially if you want to identify mushrooms. So if we need a spore print and we're throwing all our mushrooms in together, there's potential for a lot of spores to kind of mix with each other. So if you ever did need to have someone kind of look at it under the microscope, you might have a lot of cross contamination. So keeping individual mushrooms that you want to identify separate can be important, and you can wrap them in aluminum foil, or I use waxed paper bags. Um, you want to avoid plastic. It's not like the end of the world if you found a cool patch of morels and you need to bring them home and all you have is a Walmart bag. It's not like they're instantly going to go bad. But if you store them in plastic, they will go bad. They're mostly water. So if you trap all that moisture in there, it just speeds up the decomposition. The aluminum foil and paper bags, you kind of let them breathe a little bit more while still keeping them separate. And once you have all these individual things, you can't really carry them like this. So another important thing to bring is an adorable wicker basket. I have a bunch of them. If you go on walks with me, you'll see my wicker baskets. But yeah, you generally see folks carry their mushroom collection in wicker baskets, especially if they're out foraging like a specific species, or if, in my case, if it's like a foray where I'm just trying to identify some, I'll have this full of like individually wrapped mushrooms. And the wicker basket kind of helps make sure airflow is still consistent so these mushrooms aren't getting trapped in their own moisture. I've also seen people use mesh cloth bags like camper bags fairly often. You also want a knife or a digging tool. This is something I generally talk more about in the field as opposed to online, but you need to dig down. So like in this case, this mushroom would actually be really hard to ID if I just picked it off where it met the ground. So you want to get in the habit of always digging down because now, with this incredibly long stipe that almost looks like a taproot, this actually becomes very easy to identify. But without that taproot, you'll probably end up just calling this an LBM or a little brown mushroom. There's also structures on some highly toxic mushrooms, amanitas, called a volva, this little broken egg structure here. If it ended right here, right at the ground, and I just chopped it, I would never know that this had that structure, and this would become really hard to ID. Or worst case, if I mis-ID'd it and ate this, this could be the end. So digging down, I will end up seeing that structure. So you do want a knife that you don't care if it gets dull, or a special digging tool. You also want a tree ID. Oh, well, I guess I skipped binocular and hand lens. Well, you do want a binocular or hand lens like a jeweler's loupe. Binoculars, I do recommend just because it can save some time. So you're out walking through the woods, and generally when I'm looking for mushrooms, I'm also doing other naturey stuff. So I'm birding, but if I look off in the distance, like, oh, cool, what's that orange? Is that some chicken of the woods? I can bring my binoculars up and quickly realize that it's just a big orange sycamore leaf and not walk all the way through to find out and get disappointed. So it can save you some time identifying trash or very colorful leaves that look fungus like. Hand lenses are like jeweler loupes. They can help you look at those microscopic or near microscopic characteristics like the fine velvety hairs on turkey tails, or the scales that might have rubbed off on some of the mushrooms, or striations on the stalk of the mushroom. So hand lenses are really useful. And just so you know, if you're ever out and you need hand lenses, so just take your binoculars and look through one barrel opposite how you normally would, and that'll act as a hand lens in a tight spot. So that will give you, in this case, it would be 10 times magnification. I also put in tree ID book. If you're not already really good with trees, it's very, very helpful to know your trees when you want to identify mushrooms. Because, for instance, with that cracked cap polypore, knowing what a black locust tree is, you can identify that one very easily. But also, some mushrooms are mycorrhizal associations with oaks. So knowing that oaks are nearby can help you. Some mushrooms will only grow under pine trees versus spruce trees. So knowing your trees is very crucial. And sometimes just keeping a little pocket tree finder, I really like this little pocket field guide, in your pack can at least help you figure out, am I looking at a beech tree or am I looking at an oak tree, if you don't already have that. Which, by the way, we'll do winter tree ID walks. Keep an eye on the calendar. I think they're scheduled for November. And just your normal hiking gear stuff. Just you're going out in the field. Sometimes you're going to go off trail to take a look at some mushrooms. So just be prepared for whatever that might bring. So this is generally what I recommend whenever you're going out looking for mushrooms. And to continue on with the resources, I usually get asked, well, what about books? So there's three that I ended up putting on this. The first one is Mushrooms Demystified by David Aurora. This is a big book. This is actually this is the book that I got in my first mycology class and was the textbook for the class. I really do enjoy it. It's outdated, but that can be said about basically any mushroom field guide. And we'll talk about how you can kind of offset that. But this is all dichotomous keys with some species information. So dichotomous keys are those, um, is it growing on or near charred soil? Yes, go to that number, or not typically, go to this number. So it's a lot of keys that kind of help you out. And then anything by Rudy here is generally good, but I do enjoy the mushrooms of West Virginia and Central Appalachia. It's not quite something I want to keep with me in my backpack, but I usually have it in my car. It's got some really good species descriptions as well as similar lookalike. It's sorted out pretty well. It's sorted kind of by the categories we talked about today. And then my most recent one, this is a lot of my mushroom books are tied to the Southeast, and this is the first one I bought for the Mid-Atlantic. But I like this one. This one actually is more of a field guide. So this one I wouldn't mind having in my backpack. And this goes interestingly enough by season. So you have spring, summer, fall, and winter mushrooms, what you most likely see during those seasons. But he also does do a good job at the beginning of showing some of the identifiers that we talked about. So those are the three books. I'll put those in the email follow up tomorrow morning. And I also can't recommend enough the Western Pennsylvania Mushroom Club. So they meet, they have monthly meetings. I believe they meet at Frick and an environmental center. So you can find out more at their website, but they'll do so they're called forays when you're out looking to identify mushrooms and not necessarily to eat mushrooms, but they'll do forays open to the public. They'll work with a group like Allegheny Land Trust to do different walks. So highly, highly recommend getting involved with them. You'll gain so much knowledge. Any of these types of mushroom clubs, just a wealth of knowledge. Whenever you go to identify stuff, iNaturalist can be helpful. There's also a website dedicated to citizen science mushroom observations called Mushroom Observer. Mushroom Facebook groups can also be very helpful. In fact, my wife is a veterinarian, and if a dog gets poisoned by a mushroom, they don't, when they call poison control usually is going to say, post a picture on the local mushroom Facebook group. Because you got a lot of people on there that they love to identify stuff. There's also professional mycologists on there or amateur mycologists that are professional knowledge level. So mushroom Facebook groups can also be a good source. With any of these, don't get discouraged. Sometimes I think things get lost in translation when comments are made. That makes it seem like people aren't being very helpful or accepting of the fact that some people are novices. Don't ever feel like that. Just go with it. Sometimes tone doesn't translate in text. With any of these posting things, don't do this. Don't just post one picture and say, "Can I eat this?" Just don't. Definitely multiple pictures from different angles. Pick one, get that underside. Maybe even go home and get a spore print as well that you can post. Give a lot of information. So, like in this case, gregarious, or there's like multiple, all growing on leaf litter inside a rotting sycamore tree. And the forest was overall an oak hickory forest, so there wasn't any like pine component. All of this can help give you like one, show that you care enough to post a lot of information. And two, it can probably help them ID the more information they have. Online resources, mushroomexpert.com is pretty cool. This is not going to be a useful resource if you're into looking for edible mushrooms. He does not talk about edibility at all. It's just purely what you can see at a macroscopic level, so out in the field, and what you can see at spore or microscopic level. And then Index fungorum. Whenever whenever I identify a mushroom in this book, I know the name is probably not correct. The scientific name is not correct, but Index fungorum is just a simple website that you type in a scientific name of a mushroom, and it's going to pop up a big list with at the top the most accurate, recent, accepted scientific name. So this is when you get into like the nerdy aspects and you just want to kind of make sure that you're posting or you're trying to confirm what the current name is. Index fungorum is where you want to go. And before I open this up for questions, I just wanted to say I think I have a couple spots left. Last I checked, I had 6 spots remaining for my mushroom walk that was erroneously called a fall fungi foray. This is Saturday at 10 a.m. at Beechwood Farms. So if there's still spots remaining, you can sign up for it on our calendar events. Again, this is this Saturday, and we'll do a little walk around Beechwood. And with that, any questions? Because I'm all ears. I hope I got a laugh out of this because I was really proud of that photo. All right, I will open up the chat. All right. So I had one question of wanting to remove mushrooms from my landscape from the mulch. Is that bad? It's going to be nearly impossible because that mycelium is so prevalent. When it wants to come up, it's going to come up. I mean, it's a natural part. They're usually rotting something away. In mulch case, what it is just it's dead substrate material. Um, you can try to make sure it doesn't dry or get wet in the area, and that would kind of reduce the fruiting bodies forming. Or you can just like get super excited about the bird's nest fungus. But really, all they're doing is degrading some of that wood. Um, and oh, I'm glad. And yeah, I will send out a recording in the morning, and I'll kind of include all the resources that we talked about, and also a few links for some additional resources that some of you might want to see. All right. Well, I'll stick around for any questions. You guys should be able to unmute yourself. Otherwise, I am seeing the chat. I see now that they're going directly to me, so it's not like a group chat for whatever reason. But any questions, I will gladly take them. Yeah, and hopefully it wasn't too overwhelming. I know there's a lot, and it was a lot for me to try to figure out what is important. But it's a very exciting thing for me, so I really enjoy fungus. As dry as it has been here in Western PA, would mushroom forays be more difficult to find fruiting? But yeah, so it is. I'm I just told you all to sign up for the mushroom walk, but I'm also not expecting to see a whole lot. I did two at Todd and Suckup, and we had just gotten the rainstorm, so this was last Saturday. We just gotten some heavy rains that helped out, but yeah, you really do need some water. Drought is just awful. I've led many a foray in drought conditions, and what that ends up being is we're really going to focus on polypores. So we're going to focus on the shelf fungi. You're going to start to learn what to look for a true turkey tail versus a false turkey tail. But yeah, drought can be awful. You do want cool, wet nights to really get those fruiting bodies to come up. So yeah, someone do a rain dance for me so I get some rain. Does fungus on tree indicate anything about the health of the tree? Yes, it can. Uh, Sometimes no, though. So the wood, if it's in the wood and it's just degrading the wood was already dead at the center of the tree. But there are some. So if it's a saprotroph, it's essentially just eating the dead material of that tree. But there are some honey mushrooms that are parasitic on the live tissue and are actually affecting it. And then there's some that just are on the like outside. They're eating away at the bark and not much more. So it can vary. It depends on what the fungus is. If it's a honey mushroom, that could be problematic for the tree. If it's the cedar apple rust on an apple tree, that's definitely problematic for the tree, but it really depends on the mushroom. Is UV light useful in identification? Oh, yeah, that's a good question. So, is UV light useful in identification? Uh, like most things in science within the past decade, we are starting to discover that a lot of mushrooms fluoresce under UV light. We really discovered that a lot of amphibians also fluoresce under UV light. But yeah, there can be some identifying characteristics, and also there's just some cool stuff. So jack-o'-lantern mushrooms do look up jack-o'-lantern mushroom. So this is a toxic mushroom. Don't eat it, but they're really real bright orange, and the underside, the gills, will actually fluoresce green. They'll just glow in the dark, but some of them will fluoresce more under UV light too. So as far as identification, I don't know of any way to like definitely identify them yet. I bet you it's kind of a new thing that a lot of people are discovering. But if you have a UV light and you like posting pictures of them, I say definitely add that as well to any observation you make. Ooh, good question, Cindy. If you pull a shelf mushroom off a tree, does it harm the tree? Yes. Yes. If it's a live tree, and so for instance, Ganoderma suga. So this is that red varnish cap mushroom that we looked at. It's closely related to a mushroom called reishi. So that's Ganoderma lucidum, which is more of an Asian species. A lot of people started going after Ganoderma suga, and if you're not being ethical about it and you just go up and carve off to the base, you're going to end up carving some of the bark and potentially opening that tree up to a wound situation. So in the Smoky Mountains, for instance, if you didn't know, the Great Smoky Mountains National Park, you could forage in that national park. It was actually part of the way that park was made, was to still allow some people to forage. So you could do a pound per day per person of forageable items, and that included edible mushrooms. But they put a moratorium on shelf fungi because people were going in and ripping these Ganoderma suga off the trees and harming the hemlocks, which were already not doing too great from the hemlock woolly adelgid. Yeah, ripping off shelf fungi from a live tree can definitely expose it to harm. And will the shelf mushroom be okay to keep? So you can definitely, like, I've got polypores that I collected 10 years ago that I still pull out for programs because most of it's just wood. So there isn't a lot to like rot on its own or get moldy. But they're the shelf mushroom, if it's a woody one, generally stays. I've got turkey tails laying around somewhere that are just they look a little dried out and shriveled than when they were fresh, but they're still identifiable. But those all came from dead trees. I do want to make a note of that. How did that bug get mushroom growing on it? How did that bug get mushrooms? Oh, are you talking about the cordyceps? Like the grasshopper? Okay, yeah. If you want nightmares, look up cordyceps. So, do do do. So this is a fungus genus that generally targets arthropods, so insects, spiders, and it actually the mycelial the spores go in the air, they land on the insect, and they germinate on the insect because the material, its substrate that it needs is that insect's body, and so that spore germinates and the hyphae infiltrate it, and you actually have instances where it changes that insect's behavior and sometimes in favor of the cordyceps mushroom for ants, the ones that infect ant colonies. It actually causes them to seek a high spot up near the colony so that as the spore or the mushroom forms and germinates and the spores release, it's now high up and can rain down and create havoc on the colony. It is terrifying. Um, yeah, it's well, I think if you've ever heard of it was a video game first and then a series on HBO called Last of Us that like there was a zombie apocalypse show with mushroom faced zombies. It was their theory was that like cordyceps made the jump to humans. Anyway, they're cool looking. So another question is, has a patient that has shelf fungus growing in her lungs because of having the spores breathed in? Okay, so that is something that I've heard about. So a patient that has shelf mushrooms growing in their lungs because of having the spores breathed in. My understanding is that that can happen if you are on immunosuppressant drugs. Was that the case, or was this just like a healthy individual? Because that is some fear that some people will have is that if you breathe in these spores, are they going to cause issues in your body? And generally speaking, no, because we are not the proper substrate for the mushrooms that are growing on a log. They can't take hold in us. But I have heard of isolated instances where people that were on immunosuppressant drugs that were mushroom foragers and would kind of do the smell test because some of these mushrooms have unique smells and would smell it up and then they would end up with a an infection. So yeah, if you're immunocompromised, it's best not to like put it right up to your nose and smell them or get down and like play with a puffball. Healthy individuals, this should not be a concern. In fact, I don't want to stray on this too much because there's already a lot of like fear around mushrooms that is unwarranted. But it is definitely like a concern. Any immunocompromised person, just the spores are microscopic. Um, how does foraging or collecting mushrooms interface with efforts like leave no trace ethically? Yeah, that's a good one. I know it's a heavy question, but something I was thinking about when you mentioned digging up the portions for identification. Thanks for not judging. Just curious what the conversation is among experts. It's definitely a question. So one, I only ever do this on land that I've been given permission to. So it's either my own public or my own private land or public land that has allowed for mushroom foraging, like state game lands or state forests. Beechwood Farms is not a place that allows for mushroom collection. I can just if it's for one of the programs, but even outside the program, it gets frowned upon because we are a preserve. So there is the idea of like whenever forays go out and do this, we want to be as leave no trace as possible. Um, so there it is kind of this ethics thing. Whenever I find mushrooms, I don't eat a lot of wild mushrooms, but the ones that I do, I make sure I don't let my eyes get bigger than my stomach. I be reasonable, and even if I find like what looks like 20 pounds of chicken of the woods, which is my favorite mushroom to eat, I only take one, and I make sure to carve well away from the log. And it is this idea of like you want to leave no trace. When I dig up a mushroom, I just pack it down again. Just like if I was looking for salamanders and I kind of moved the leaf litter, I put the leaf litter back. I always try to stress those on my walks too. Like I think ethics is something that should be talked about more and more. And actually, you might hear more and more about that from our organization. We'll hopefully be doing some more like ethics out in the wild, and foraging is definitely something that we'll talk about with that. All right. Any other questions? Well, I hope you all enjoyed the presentation, and I think I will shut it down here. And hopefully, we'll be doing some more mushroom programming in the future, but also definitely check out the Western PA Mushroom Club. I mean, they're great for it. The Spirit of Ethic. Braiding Sweetgrass is a fantastic book. Yeah. Gathering Moss as well is just is a great read. Thank you all. They can just come along, Jimmy. All right, you all take care.